Welcome back. My name is Julie Grinstead. I'm a PhD student in the Wildlife and Fisheries Biology program, and Dr. James Anderson and Dr. Lydia O'Halloran are my advisors. And today I'm here to talk about banana water lily ecology, implications for waterfowl management. So banana water lily is the scientific name. It's Nymphaea mexicana, and it is an emergent aquatic plant, and it is native to the southeastern United States and Mexico. It is used as a food for waterfowl. Waterfowl like to eat the stems, leaves, flowers, seeds, rhizomes, and tubers. It also harbors macroinvertebrates like insects that they like to eat. And it's also easy to maintain and grows easily. In fact, it is such a hardy plant, it is invasive in other parts of the world like Africa and Australia. So banana water lily grows in full sun in approximately 46 centimeters of water. It flowers for up to 2 consecutive days, sometimes more, usually between the times of 11 a.m. and 4 p.m. It spreads by rhizomes, which produce stems, leaves, flowers, roots, and tubers, as well as stolon or runners, which produce roots and new plants. And this is a rhizome here in this photo, and it sort of looks like a pine cone. And each node produces a stem, and a stem will produce either a leaf or a flower. And at the end, you have roots. So this one rhizome floats in the water. It's not growing in the pond sediment. And it also produces the stolon or the runner. So this is an important structure on the plant. It also reproduces via cross-fertilization, as well as asexual propagation. So what it does is, when the flower blooms, the first day it's a female. Then the second day it's a male, and it only blooms for 2 days. During that time, it is pollinated and cross-fertilized, but it also reproduces asexually as well. And that's one of the reasons why it is spreads so quickly and is such a prolifically growing plant. Below the water surface, the rhizome, again, it's this sort of pine cone shaped structure. It produces roots, tubers, and stolon. And these down here are called the tubers, which the plant gets its name from these because they look like little bunches of bananas. And the stolon are these large white root-like structures, which are different from the roots in that they grow out from the plant and they produce more roots in another plant. Above the water surface, the rhizome, that pine cone shaped structure, will produce the stems, leaves, flowers, and seeds. And here you can see a seed pod. These can produce up to 20 seeds in one pod. And so you can tell that from just one plant, you can get up to 20 other plants through the seeds. And here is a full plant from the leaves all the way down to the stolon, the roots, and the banana tubers. So why are we doing the study? Well, we want a better scientific understanding for stakeholders such as landowners, farmers, hunters, and others regarding the productivity and reproduction of plants that are either protected or foraged. The maximum production of banana water lily is also something we want to know, and how to maintain the genetic diversity of banana water lily populations to maximize productivity. Also, how do we apply waterfowl management principles of banana water lily to inform wetland conservation? So the objectives of our study are twofold. The first is to evaluate the impact of waterfowl herbivory on banana water lily tubers, rhizomes, stems, leaves, flowers, and seeds. And our second goal is to evaluate the full potential of banana water lily growth under controlled grazing conditions. So in this, within our objectives, we would like to measure vegetation characteristics, such as percent cover, above ground and underground growth in biomass, and observe the change over time in regard to the treatment type. We also want to study the effect of soil and water characteristics, as well as climate and herbivory on the growth of pond vegetation in relation to banana water lily. We'd also like to evaluate the differences between waterfowl only grazing versus waterfowl and aquatic herbivory. So our study area is located near Georgetown at Ingleside Plantation, which is a 19-acre wetland impoundment near Georgetown. So in order to carry out our study, we determined that we use 3 treatments. So our first treatment is our control, which is basically a plot in the pond where there's no netting. There's nothing other than just some rebar in the pond sediment. And we want to use that to allow anything really to come and feed within the plot, such as waterfowl, aquatic vertebrates, and macroinvertebrates like insects. Our second treatment is a total exclosure. Okay, this is a plot that has some netting around it, and it keeps out all waterfowl, aquatic vertebrates, and it keeps out all waterfowl and aquatic vertebrates and allows macroinvertebrates to get in. And our third treatment is a partial exclosure. So there's netting around the entire plot with an opening at the bottom, which allows access to diving ducks, wetland vertebrates such as turtles and large fish, and macroinvertebrates. So here is one of our prototypes, which I built out here in front of this building last fall. It's a partial exclosure. And here you can see the rebar, which I have down in the ground. And here's the actual plot in the middle is this white square made of PVC pipe. And then the netting around the outside is meant to exclude some organisms, but not all. So we have this gap at the bottom, which would allow diving ducks to get under. And so here is a scheme of our treatment plot. So in the middle is the actual plot. Okay, this is where we'll be sampling vegetation and macroinvertebrates and soil. And around it is our buffer. And the buffer is just to keep the plot from being disturbed by outside activity. And what we used around it was like this mesh netting, maybe what you would think of as deer netting or garden netting, that kind of thing. So last fall, we set up 15 plots at 5 sites in Ingleside Plantation Pond. So within each site, we have 1 treatment of each type. So 3 plots. Here's a control plot. Here is our partial exclosure, and the orange is the total exclosure. We also have set up some cameras for some remote viewing so that we can view the waterfowl. We can see what kind of behavior they're exhibiting. We can watch them feed. And we can also do other things like measure percent cover of the banana water lily. And here I have a camera at this meter stick, which I use to measure the water level. So 15 sites, 15 plots in all. Here is the water control structure. So at this site, we are able to raise and lower water control structure doors so that we can allow water in and out. So which means that sometimes the salinity may change. We are measuring that, and we want to observe how that affects the growth of banana water lily and herbivory. So here's sort of a flow chart. It shows all of the variables that we want to study, water variables, soil, vegetation, macroinvertebrates, and waterfowl grazing. And we're going to observe all these variables in each treatment type. And the end result is data. And we're going to break down these variables into two types of variables, our explanatory variables and our response variables. How does treatment type affect the growth of banana water lily? Do we find our soil vegetation variables altered from different types of treatments? Do we find that different communities of macroinvertebrates affect the waterfowl that visit those plots? So we want to try to develop an understanding of the relationships between the treatment type that we used and our variables. So in trying to carry out our study, we have chosen a quantitative data acquisition, which means that we are going to be counting and using continuous data to compile our sample in a systematic random sample design. And what that means is, you go back to our map, our sites are set up in a systematic way, but the treatment type was chosen in a random fashion. And so that's why we're calling it systematic and random. And we're using this in an experimental research design. Our statistical analysis will be looking at descriptive statistics like the mean, the median, the standard deviation, the variance. We'll also be using scatter plots, histograms, bar charts, box plots, and graphs. We want to be able to analyze the normality of our data, any relationships, and we'll try to draw correlations and from there develop a model. So any results we've obtained so far, we have seen that we've observed the growth of leaves in the treatment plots to be higher in the total exposures. So this is not unexpected. In our total exposure plots, what we have is a plot where no organisms, no waterfowl, no macroinvertebrates are allowed to enter. So we have more growth of banana water lily. And our control plots, surprisingly, are a little higher than the partial exposure plots, but over time we might see that change. We also have found that when we examine our soil vegetation, which is the banana tubers, our maximum number of banana tubers in one plot, in one sample, we were able to extrapolate that out to 1 acre. And we determined that you could grow 18,749.94 pounds per acre of banana water lily tubers. Now, the tubers are just one way that this plant reproduces. And that means that in addition to the seeds, in addition to the tubers, and I'm sorry, the runners and stolon, that this plant is extremely highly reproductive. In hectares, it's 21,027.60 kilograms per hectare. And the average would be 6,005.37 pounds per acre, or 6,734.99 kilograms per hectare. And the same information we have used in a graph or a bar chart, which just shows that for pounds per acre, so far our numbers are showing that our partial exposure plots are producing the most soil banana tubers. Now, the banana tubers are sort of like hibernating bodies. They grow down into the soil and they can stay down there until ideal conditions trigger their growth. So when I say that partial exposures are producing more of these soil banana tubers, it could be that over, say, the course of time, that these are very initial results. So over the course of time. Now, above the surface of the water, what we can see from last fall was at the beginning of fall migration in November, and the waterfowl fly south around the middle of October. You can see lots of banana water lily out here on the pond. Only 2 weeks later, 3 weeks later, you can see how many waterfowl have arrived. And by January 31st, the same camera, the same plot, you hardly see any banana water lily at all. So the results are significant, and that we can see that banana water lily is definitely being grazed upon and is definitely a food source for waterfowl organisms. In addition to our plots, we have a flower square, and this is a 10-foot PVC pipe. It just floats out in the water, and back in April, this is how much banana water lily was growing there. And by June, it had filled in. And every day we look at the camera photos and we count flowers, and we watch what time they bloom and what time they close again, and how many days they last. And in that way, we can determine flowering times, peak blooming, and the number of blooms per day. So given all this information, we hope that it can provide insight for researchers and stakeholders. We want to understand the reason for the plant's success. We'd like to utilize banana water lily as a cost-effective, manageable food source. Okay, so from a agricultural standpoint, we're thinking about stakeholders like farmers, hunters, and landowners. Planting banana water lily is a very viable option because it's easy to grow. It grows quickly. It spreads easily. It is very popular with waterfowl, and it has several mechanisms for reproducing, which make it a very cost-effective strategy. We'd also like to learn, We'd like to learn the underlying reasons for its success and how to optimize the favorable characteristics of this plant for efficient use as a waterfowl management tool. We'd like to thank Jim Hills, the owner of Ingleside Plantation, for all his help in this project. And I'd like to thank my advisors, Dr. Jim Anderson and Dr. Lydia O'Halloran, for all their great advice. And thank you for listening. Questions?
I have more questions than time allows. Yeah, I will definitely do that. Challenge that may be. First of all, your study is on Ingleside only, it's 19 acres. So it's a single site, right?
So far, yes.
So far. See, the history of this pond, management. Was it a tidal and okay. So you've experienced some cement
as high as 24 parts per thousand.
Right.
But I And outside, and we had an explosion to keep out turtles. And then, here's what's funny part is, it's those banana tubers, we found some of those, put an explosion in. Boot went down, something white, pulled it up, looked at it. Those things started sprouting, and in about a month, the whole pond came back like nothing ever happened.
I think it experienced the same thing on Blue Isle. Yeah. Cape Romaine, with storm events and that kind of thing.
Uh-huh.
And freshman effect, which sort of brings me to kind of my main point. Have you, or would it be possible to consider not the intensity of the study or the inside, but just general occurrence, say, in the coastal zone and looking at ecological factors?
Yes, no, we
Both
definitely,
physical and biological.
no, we
And
definitely,
in a broad sense, looking at the whole community, you know, what other species occur, what other species have plants, SAB, and that kind of thing. But not the intensity of UV, exposures, et cetera. It's you doing the angle side, but I think it'd be very interesting since this is a PhD level project to look at some other sites, do just a quick survey.
well, we'd love to do that. Are you going to let us come to Bull Island?